Welcome to the UK Pharmacogenetics Stratified Medicine Network meeting. It's our 11th annual meeting. And again, whenever we have this meeting, it's always sunny outside. So somebody must know that we have a meeting on a particular day. So I'm just going to give you a few brief opening slides and then go to our opening plenary session. So as you know, over the last year, since the beginning of this year, the UK Pharmacogenetics Stratified Medicine Network has been supported by the British Pharmacological Society, and I want to thank the British Pharmacological Society for supporting the network. Even though it is supported by the BPS, we did discuss this, and we've kept the original branding. So the original branding still stands. The purpose remains the same, and the objectives remain the same for this particular network. So it has three main objectives. First is to serve as a vehicle to showcase advances in the whole area of personalized precision medicine, which is vast. And you'll hear many of the talks today in some of the areas, but we've covered many different areas previously in this conference on precision medicine. So it is vast and so on, and it's good to hear about the advances. And I don't know of another conference that covers the area of precision medicine every year in the UK. Obviously, it's good to have the advances and get good publications in nice journals. But the most important thing is how can you actually improve the uptake of these technologies into clinical practice for patient benefit. And I think there's a lot of expertise, and this group is very much interdisciplinary. There's a lot of expertise in the audience, and we need to be able to learn from each other how we can actually help that innovation, uptake, diffusion into the health service. And obviously, one of the main reasons for setting up network was to act as a vehicle for people to be able to network with each other, collaborate, and really develop, hopefully, grants and so on. And so it is really important that we continue to keep this as an interdisciplinary membership network, and it remains free to join. So please do join if you are not a member. So we've had a busy year so far. We started in March this year working with the Pharmacogenomic Medicines Optimization NHS Genomic Network of Excellence, which is funded via the Genomic Medicine Service, and you'll hear from Sue Hill very soon. And this network is led by Bill Newman, and so we held a very successful meeting in Liverpool at the Spine, which is the RCP UK, RCP North headquarters. And there will be a paper coming out from this, hopefully in the near future. And that was, again, attended by over 100 people. We've also been very successful in obtaining funding from Innovate UK for a regulatory science and innovation network in pharmacogenomics. And for the last few months, we've been busy really working on that area. It's a very complicated ecosystem in terms of pharmacogenomics and the regulatory aspects. And we've identified 4 different challenge areas about pharmacogenomics information, drug labels, and developing guidelines. And I feel that as we move forward, developing guidelines which are UK-specific is going to be a key focus of how we move forward. The other aspect, the challenge area 2, was embedding pharmacogenomic information in electronic health records and the clinical decision support systems. And they need to be intelligent. If you have an unintelligent clinical decision support system, people would just switch it off and so on. So we need to think about how we can develop that. And we had a very nice workshop last week on how do you regulate genetic diagnostic tests. And it's a very complex ecosystem, again, with some companies having to follow 15 different standards to be able to adhere to all the regulations and so on. And next week we have a cost-effectiveness approach in terms of pharmacogenomics. And the aim of this network is to really develop proportionate, forward-looking, inclusive and responsive regulatory pathways that enhance innovation. So we've held 3 workshops so far. We've got a workshop on health economics next week, and we've then got an interdisciplinary workshop on the 1st of August. That will be at the Royal College of Pathologists in London. So we are in the discovery phase at the moment, really understanding where the barriers are to innovation, barriers to regulation. And we want to learn from different people in this area. And what we're then planning is the implementation phase. Again, going to Innovate UK for further funding to see how we can develop the solutions. And so really the workshop on the 1st of August will be really discussing how we develop those pathways to be able to improve things for regulation. So upcoming events, we've got a joint webinar with Association of Physicians on the 3rd of October. Again, that will be free, so please do try to join that. We also have a symposium on pharmacogenomics of adverse drug reactions at the British Pharmacological Society annual meeting, Harrogate, 12th of December. So please come to the symposium, and then you can go to Betty's Tea Shop if you want afterwards, but don't before. Focus this year has been on pharmacogenomics, largely to embed the network within the British Pharmacological Society. But as I said, we cover all aspects of personalized medicine, precision medicine, and this is evidenced by today's program. And we do want to cover other areas of precision medicine as we move further and forward in the future. So if there are particular areas you feel that we should cover, then please do get in touch. My email's on the side. Happy to have a chat and listen in terms of what we can cover for the next year. So I want to thank all the sponsors for this event. They're all upstairs. They've got stands upstairs and so on. So please do go and visit the tables and so on. And thank you for attending today. And what I'm going to do now is to go straight into our first plenary session, which is what you're here to hear really, in terms of the speakers. We've got a fantastic line of speakers this morning. Starting with Professor Dame Sue Hill, who doesn't need an introduction, really. She's Chief Scientific Officer for NHS England. Sue has done many different things and is the architect of the genomic medicine service infrastructure that we have in the country at the moment. And Sue is going to tell us on accelerating genomic medicine in the NHS. So, thank you.
Thank you very much, Munir. I'm delighted to be back at this meeting, and it's great to see the progress that's been made since I came to one of the first meetings, I think, Munir. I'm going to really set out in the next 20 minutes how we've been putting into place the infrastructure for accelerating genomic medicine in the NHS. And by genomic medicine, I don't just mean genomic testing, I mean the end-to-end pathways. And I think as we consider pharmacogenomics or stratified medicine, we need to ensure that it embraces that end-to-end. So I want to start by setting out that in 2018, we launched the NHS Genomic Medicine Service, and this was to provide equitable access to all eligible patients in the NHS in England, so some 55 million people. Key to this was the establishment of 7 NHS genomic laboratories as hub laboratories. To wrap that around with 7 genomic medicine service alliances where we funded a multi-professional infrastructure and leadership infrastructure, including of pharmacists and general practitioners. And then 17 clinical genomic services that really are our key clinical service that supports rare and inherited disease, but also supports some of the testing from a clinical perspective of somatic testing in cancer. We've recently established cellular pathology genomic centres, recognising we needed to streamline the pathway from tissue acquisition in cancer through to genomic testing if we want to stratify medicines, chemotherapy agents, early on in the patient pathway as first-line agents. And then we have a genomics education programme that is looking at the workforce, and then a partnership with Genomics England in the delivery of our whole genome sequence service that still remains world-leading in terms of offering whole genome sequencing from a routine perspective, focused on some cancers, on some rare diseases, at the moment. So these are some of the areas that we wanted to ensure that we created in within this genomic medicine service. We have a national NHS genomics unit that sits within NHS England that coordinates, sets the direction, standardises, commissions and contracts, because all of the money for this service that since we introduced it in 2018 has had over a tripling in terms of the investment is under the control of the genomics unit in NHS England. But you can see that a key part of what we do is making sure that the service is informed by our patients, our users, our communities, but that we use a data-driven led insights approach, and we align research and development with our routine care within the NHS. The key offer for patients within the accessing the NHS in England is our National Genomic Test Directory. And this again still remains something that is world leading. And this focuses on rare and inherited disease, on cancer, and also on some pharmacogenomic applications. And we expect those to expand over the coming years. It covers testing from single gene right up to whole genome sequencing in terms of who's eligible and the type of technology that should be applied, rather than specifying, for example, a particular commercial application. This test directory and the evidence that is associated with introducing, let's say, new genes, changes to the eligibility criteria, new mutations is reviewed annually, but we also have the ability to fast-track applications, which responds to, for example, new licensing of medicines, such as olaparib, which we were able to do in year. Since the test directory came into being in 2018, we've made 350 different updates. And the area that we're rapidly updating is always in the area of stratified medicines, so new precision medicines, and the genomic targets that are associated with that. But also some of the emerging findings from diagnostic discovery, still from projects such as the 100,000 Genomes project, that changes maybe what we want to test or the eligibility. In 2022, we published our first NHS genomic strategy, which link with, these different strategies on the right-hand side here, including the NHS long-term plan. Of course, with the new government, there will be a new direction potentially in some of these areas. But if you read the manifesto, you know that there's a commitment to the life sciences in general, certainly in the two big parties. Our key themes in this strategy, which set out 36 commitments to be delivered over a 5-year period, are around embedding genomics in the NHS, delivering equitable testing, and enabling precision medicine and reducing adverse drug reactions, enabling genomics to be at the forefront of the data and digital revolution, understanding that there's a lot of informatics developments that still need to happen to make this a reality in the system. And Munir just alluded to this in terms of some of the work that needs to be done around decision support, for example, to implement pharmacogenomics in a widespread way across the NHS. And our fourth area is evolving the service by cutting-edge science, research and innovation. And I'm going to go through each of the 4 priorities pretty quickly. So this is our priority 1 for embedding genomics in the NHS. What we've been able to do is create integrated services such that we have 7 regional genomic medicine services that are led by chief execs in the system. We funded the leadership, as I mentioned earlier, that has been really important to pull those posts out to make sure that they could focus on developing the infrastructure, embedding genomics. We now publish quarterly activity data and will begin to publish turnaround times. I've already mentioned we've established cellular pathology genomic centres, but we've been supporting a broad workforce development programme, including in pharmacy, which is to develop that workforce really to support us in the approach to introducing stratified medicines, but also in pharmacogenomics. We have national collaboration with many different countries who all want to learn from what we're doing in the NHS. One area we have been focusing on is establishing an ethics, equity and legal advisory board. You can see the people who are part of this board on the left-hand side here. And we really want to have one place where we can consider those ethical and legal themes that are emerging within the genomic medicine service, but also a mechanism to really understanding, are we delivering an equitable service to all patients that need it in our diverse communities. And we've currently partnered with the NHS Race and Health Observatory to oversee some research on identifying and assessing inequalities. We're starting on the inequalities relating to testing, but over time we will start to look, for example, at the inequalities that might exist in terms of access to a stratified medicine. So this is a start of a process really looking systematically at inequalities by the use of data. Our second priority is about delivering equitable genomic testing in that end-to-end pathway. You can see our priorities at the top here, which I won't sort of go through, but just focus on key areas of progress. There's been a year-on-year increase in the growth of genomic testing that we're doing in both rare and inherited disease as unmet need gets released, and in cancer, and in a small number of pharmacogenomic applications, such that we're now at over 800,000 genomic tests being performed per year, with a significant growth planned for this current financial year. And as part of our whole genome sequence service, we've now sequenced over 100,000 whole genome equivalents in a relatively short period of time. And whole genome sequencing will be a part of the future of the NHS Genomic Medicine Service. And we're currently looking at the model for doing this in the future in terms of looking at a transition from what we do now with large panel testing, for example, through to what we could do with whole genome sequencing that gives us other genomic data that can be actionable. The key point of this is making sure that we get the price point right and we have the ability to analyse. Since between 21 and 23, we've delivered over 1 million cancer genomic tests. And cancer genomic testing didn't come into the genomic medicine service. So in our previous genetic laboratory service, they didn't test cancer tissue. But when we established it in 2018, it did include cancer. Between these 2 years, over 1 million tests have been delivered and nearly 250,000 next generation large pan-cancer panels that enables us to look beyond a single mutation and a single cancer into mutations that may be actionable by agents such as NTRK inhibitors. We've been working really closely with the experimental cancer medicine centres, with our commercial medicines unit, with NICE and with industry to try and align clinical trial targets with standard of care so that they would be part of the panels that are run within the genomic laboratory hubs, which will enable a really rapid access to clinical trials without additional testing being done or needed to be funded separately. And we're working to implement nationally agreed turnaround times in cancer. And this is the first time nationally agreed turnaround times have been agreed across all the major cancer types. which gives us a challenge because for most of these, this is about delivering from tissue acquisition to a genomic result to inform a stratified medicine 14 days. So that includes through histopathology into extensive gene panel testing within our genomic laboratory hubs. We've been working with NICE and Vicky Chaplin, who's part of our genomics unit team and a pharmacist by background, has been working really closely with NICE and colleagues there to establish a genomic medtech pathway. And we've been linking this with an NHS medicines optimization board that I'm going to come back to. The work with NICE is really important because it does enable us to think about guidance and guidance that needs to have genomic testing in it and the link to a stratified medicine or a pharmacogenomic sort of test profile. So that will be, we're reviewing what we need to do and what needs to change. And we've been undertaking a series of other transformation projects to evolve the system. This is one of those where we've been looking at circulating tumour DNA, and it started with a collaboration between the Royal Marsden and Guardant, looking at the use of this test in non-small cell lung cancer. And this has since transformed the way we're thinking about circulating tumour DNA testing because a technology transfer has happened and we've been funding, that's we in NHS England, circulating tumour DNA testing in non-small cell lung cancer, and we will be funding up to 10,000 tests over this year. And through the Network of Excellence, as you'll hear from me later, we're looking at other cancer types. We see this as a key part of the development for the future because it cuts down the time until a genomic result. And in non-small cell lung cancer, it's meant that patients who haven't been able to have a bronchoscopy to get a tissue sample have been able to be tested and then for stratified medicines to be introduced. Significant health economic benefits have been demonstrated, as well as the reduction in demand for other diagnostics. So this will start to bring more genomic testing to patients. The second area I just wanted to touch on is something that I've been working really closely with the Chief Pharmaceutical Officer, supported by Vicky Chaplin, which is to introduce a genomics-informed medicines optimization board. It brings together a raft of representatives in NHS England, including from commercial medicine, from our genomic medicine regions, including all the chief pharmacists that have been appointed in all our 7 GMS regions, but also with the people involved in regional medicines optimization committees. And we've got 4 main aims. One is to ensure equitable and appropriate access to testing to enable access to new and existing medicines that are informed by genomics. The second is facilitating the development of new medicines. The third is the safe use, and then to incorporate that into the medicines governance framework. And the fourth is integrating genomics into existing treatment pathways. At our last board, we, for example, talked about introducing dosing guidance, which is a really key part of how we start to drive this and implement a number of changes. Our third priority is around enabling genomics to be at the forefront of the data and digital developments. You can see these are our 3 main areas of focus at the moment. One is to develop a digitised test directory and to introduce that over the next year if we can. The second is better coordination and interoperability within the system, adopting all of those standards for genomic data and the sharing of data. And the third, which I'm just going to come on to, is creating a unified genomic record. But you can see under our key areas of progress, we've been working in Bill Newman, John McDermott and the team in the Northwest have been really critical to us thinking about what a pharmacogenomic decision support system might look like for prescribing and for implementation across the NHS. So this is as we move to a unified genomic record. On the left-hand side sets out where we actually are, which is mainly with a paper-based system, where there's the potential for errors, difficult to manage consent, no visibility of the status of samples, and also a lack of information to drive management, unstructured data, and the inability to scale. And if we're thinking about this, particularly in pharmacogenomics scale, we're talking about hundreds of thousands of tests. We're not talking about just where we are now with genomic testing. So we've got to have a system that enables us to scale. So we'll move to something that is much more consistent for ordering and test management, driven by standards, with a modern API framework, which you won't be able to see up here, but it links to this part of this diagram. And then up there are the trusted research environments, so often referred as secure data environments, that's been set out in all the data work. But then this API framework will support integration into electronic health records. It will enable structured data, which is really important, because at the back end of the UGR, we'll be able to provide access for researchers through defined consent and other processes. Now, I just want to show, this is a really busy slide, but I just want to show you that for that UGR and its development, so from testing up to being able to access bioinformatic pipelines, to be able to store results and raw data, and then to provide access to researchers. In terms of the development of this, we're using pharmacogenomics as a case study, and you can see the different elements that are associated with this, and the link with the network of excellence in the North, led by the Northwest, that is the progress study, but much more with all the work packages you'll hear from John McDermott later. But then we're also looking at cancer and also inherited cardiac conditions in our use cases as this develops, and we expect this to be introduced in the next 18 months. Our final priority is about evolving the service through research. So one of those areas is these 8 networks of excellence that I'll come back to. We're working with Genomics England on a number of different initiatives, including the Generation Study. And we're starting to evolve something that we created before called the NHS Genomic Research Collaborative in conjunction with NIHR, with Genomics England, with the NHS Genomic Medicine Service, to essentially approve projects for access to the NHS GMS for data, for cohorts, and for other analysis. And so one of the areas that we've been aligning the NHS Genomic Medicine Service with is the cancer vaccines launchpad that was announced over a year ago. The trials in active recruitment at the moment. But essentially what I wanted to show is that these are all the elements, sorry, let me just go back a minute. These are the elements here that are associated with the Genomic Medicine Service infrastructure, the Cellular Pathology Centre, the GLHs. And at the moment, for the messenger RNA trials that are underway with BioNTech, the tissue and blood are sent to Germany for BioNTech to sequence and then to create the individualized personal vaccine. We're working at the moment to verify the ability of the Genomic Medicine Service to extract nucleic acids to the standards that are required and to undertake exome and RNA sequencing, such that all of this will be done within the NHS Genomic Medicine Service, working with clinical teams here. And then it's just the data that will be sent out to BioNTech in Germany at the moment before they set a facility up in the UK to create the individual vaccine. What underpins this is a data infrastructure. And I've also put this in to say we're working also to use this pathway for other immunotherapies in cancer, so not just messenger RNA, so it's a platform for that, and to use this as a template for a new rare diseases therapies launchpad, which brings with different types of challenges, especially if we look at antisense oligonucleotides, where we're talking about very small numbers of patients. The second area is the genomic networks of excellence, where what we were trying to do with committing NHS England money is to leverage the funding from industry or from other research bodies that have already been granted in areas of unmet need to generate the evidence for rapid adoption within a 2-year period into the NHS. And so these are the people that it brings together. And it's really focusing on translation to adoption, providing us with the evidence for diffusion through commissioning. These are the 8 areas that we're focusing on. You can see those circulating tumour biomarker testing, a rapid hemato-oncology development to support really early access to stratified medicines in hem-onc, but across a number of different areas, and I don't have time to talk here about infectious disease today, but this is a respiratory metagenomics network of excellence that's looking at sequencing the secretions from patients with severe respiratory infections in intensive care units, and within a 7 to 10 hour period, being able to target the appropriate antibiotic. Because as well as the sequencing, we understand the resistance mechanism. This has shaved off 2 days, for example, from people's stay in intensive care units, and this is being rolled out. But you can see one of these other networks is in pharmacogenomics and medicines optimization, and in genomics AI. I wanted to just say something about the generation study, which is the study that's been undertaken by Genomics England, looking at essentially more than 200 conditions in newborn babies to really detect rare disease early. And all of these conditions can be treated by the NHS through a commissioned service. These are some of the examples of conditions within that list. And this recruitment is now live in centres across the NHS with the plan to recruit 100,000 new babies. But what I wanted to show you is that what it's led is because we don't really have defined treatment pathways for babies where a rare disease is being diagnosed by a genomic test. And so through consensus, we've had to develop clinical pathways for all of the conditions that are being included, but also what type of diagnostics need to be done here after there's a positive, so here, but the type of treatments that need to be instituted. And these are treatments that generally often don't have a license for use in newborn babies. So it's been a really interesting development. Now, as we look towards the future, this is where we're heading in terms of our overall commitments, our overall strategic approach, as we're looking with a new government coming into place, with our, for example, our developments associated with the spending review bid for the future. We want to make genomics commonplace, and so we want to really rapidly accelerate mainstream. We want to deliver high throughput and rapid turnaround times in an industrialized process. We want to have a comprehensive genomic testing offering, including elements of the functional genomic pathway like RNA, like looking at the transcriptome, the proteome, but also recognising we need to significantly expand our testing in pharmacogenomic, especially with new sort of indications coming down the line, such as in the use of clopidogrel or in the new Alzheimer's drug. We will need to drive new models of care that will reduce the use of diagnostics in other types of diagnostics, but focus on the end-to-end. Integrate and use data for multiple purposes and make the NHS and continue to make it a critical enabler of innovation and research. And this will enable us to continue to be world-leading and embedding genomic medicine into the NHS. And I'll just finish with this summary, which I'm not going to read out, and say thank you very much.
Great, thank you very much. Are you okay to take questions?
Yes, yeah.
Any questions for Sue? So maybe I can start off, Sue. You talked about scaling up pharmacogenomics. You know, if you consider that every year maybe 250,000 people get started on SSRI for depression. Yeah. So that's a huge number of tests and so on, and they're scaling up in many different aspects. So can you give us an idea of what you've been thinking about in that area in terms of scaling up?
Do you mean in terms of scale?
Yes.
I mean, we're talking, I think, Munir, at sort of certainly over 600,000 tests per year. And I think we're talking yesterday with Bill and with John McDermott about, you know, this balance between preemptive testing, what that might look like, but also the reactive testing for agents such as SSRIs or, for example, some of the new Alzheimer's drugs or clopidogrel. So I think as we move forward, it will be a balance. I think our starting point will be in those where we know we need to do it, or there's a nice guidance associated with it, as we start to learn, from example, the progress study that Bill and John are doing, and you'll hear from John later, which is really looking at testing in primary care for those commonly used agents. If you ask me, as sitting within a commissioning organisation where the drug bill for some of these are going up and there's lots of adverse drug reactions, then that might be where a system might want it, but we've got to focus on also the here and now. But significant, and I think we'll be heading actually over 1 million tests in the not too distant future, just in this area.
Okay, any more questions? So we've still got 30 seconds. Maybe I can ask you another question. Oh, there you go.
Sorry. Yeah, thanks, Sue. So, Rob Hastings. I was just wondering, a kind of more general question in terms of how you think we're doing in terms of the genomic medicine moving to be a true network. I guess a lot of these places, you know, the kind of different parts have been introduced at different times, but how is it actually coming together in your mind, and what are you kind of most proud of but think there is most to do on?
So I think, Rob, it's really nice to see you again. Where we've got to with the network and working as a network is some of the GLHs, you know, a small number might do testing for the whole country. And so it's starting to evolve as a network. I think that's the area we will be pushing forward in over the next 5 years. But I think you'll hear from John McDermott later, though, as the Network of Excellence has really driven, in some of the projects we've been doing, this network approach. So sending or gathering samples, processing them in a particular genomic laboratory hub, and then moving them, for example, to the Marsden for the circulating tumour DNA testing. So it's coming together, and you know what we're learning is those centres that can do this, where there's a real commitment to investment, but also the expertise, and others that are going to need a little bit more help on this journey.
Great, thank you very much, Steve. Okay, great pleasure to welcome Professor Nick Wareham. Nick is director of the MRC Epidemiology Unit in Cambridge, and I heard him give a talk on the changing landscape of cohort studies in the UK. I thought it'd be perfect for this particular conference. So Nick, thank you for agreeing to do this.
Thanks very much, Munir. It's great to be here. You're going to hear from Raghib shortly about a very exciting new development in the UK, the start of the Our Future Health Study, which is enormous and is very inclusive and has great diversity. But if we want to use cohort studies for science, we need scale, we need diversity, but we also need elapsed time. And that will come with Our Future Health. But I want to summarize today some thoughts about how we can use existing resources, perhaps in the window of opportunity before that elapsed time develops in OFH, because I think it will. So I'm chairman, I've sort of said, of MRC Population Health Sciences Group, which is the body within MRC responsible for developing and overseeing its investments in population health science, including cohort studies. And in 2013, the MRC was 100 years old, and it looked back at what it had done in terms of population cohort studies and looked forward in terms of thinking about some of the challenges. And I want to use that as a base for thinking about how have we done over the last decade. And I guess the first question is, why is that relevant to a conference, a meeting about pharmacogenetics and stratified medicine? So I think the best way to articulate that is to read the foreword, which was written by John Saville, which is on my phone. So he said, participants from UK cohort studies have given consent for their personal data to be linked to NHS records and other data sources, such as education and the census. Linking cohort study data to routine health and administrative data sets in safe environments that protect confidentiality increases the scope and scale of research possibilities. All true. Investments from the MRC and partners to create the FAR Institute of Health Informatics Research and UK Health Informatics Research Network, brackets, which subsequently became Health Data Research UK, will facilitate data linkage to identify the causes of disease, develop personalized treatments, and monitor health risks and drug safety. Trustworthy use of personal health data by participating in studies or through use of anonymized health records is fundamental to improving care and public health and is essential for the UK to remain at the forefront of medical research. True then, true now. So how are we doing? So let's look back at some of the things that were in that report, and that was led by one of my predecessors as chair of PHSG, Jill Pell, to whom a lot of credit goes for initiating this. So we got to think about new cohorts, investments like OFH, not just in terms of scale, but also in terms of the age of the participants and also the time period in which they've lived. There has been, and I've heard it said, that if we invest in a cohort now, that's investment done, big tick, and that's it forever. But of course, we all live through different time periods, and we will need to have recruitment of people into cohorts indefinitely because we need to study contemporary health issues with a view to thinking about the future. And the fundamental problem about cohort studies, because of the problem of elapsed time, is they fundamentally telling you about the past. So we need to be forward-looking about where are the gaps. So if we look at cohorts by the start date of initial data collection, you can see why the MRC was so proud of its long tradition. And the UK is really at the forefront of building population cohort studies. It goes back to the 1930s, a few studies there in the 30s. I think the Hertfordshire cohort study, Boyd-Ord, and the Lothian birth cohort. And then in the 40s, you get the 1946 birth cohort. And you can see in the 90s into the 2000s, an expansion in the number. Now, we haven't done a review since, but I bet if you did, there'd be a further expansion in the number of cohorts, even further from that in the 2010s and onwards. So there's a long and a proud tradition of this that goes back a long way. But obviously, those cohorts started recruiting people at different ages. So some of them you can see are birth cohorts, so they start at 0. Many of them, including the one I lead, Epic Norfolk, start in midlife. And that's largely because if you recruit in midlife and you wait 10, 20 years for follow-up, you maximize the number of hard clinical endpoints. If you start at birth, studying health endpoints, unless you wait a long time, like the 1946 birth cohort, is very challenging. But you can see there's a bit of a gap in the number of cohorts that start in the teens and in the 20s through to the early 30s. And we have started one called Fenland, which deliberately started at 30, because if you start at 40 or 50, by that time, because of the amount of screening that's going on, many people already have therapies largely for metabolic disease. So if you're interested in that, you have to start a bit earlier. If we look then, this is a distribution at the top in men and at the bottom in women, and it excludes the Million Women study in UK Biobank because they just skew all the figures. But this is the distribution of the age of the participants in 2012 when this survey was done. So you can see there's a preponderance of women in studies, fewer men. And then there are predominantly middle-aged to older people. My definition of that changes as I get older. We'll come back to that because this was 10 years ago. So if you add 10 to all of these numbers, you can see that quite a lot of the participants in cohort studies now are getting quite elderly. And there is a second peak here in childhood. But again, what I said earlier, very few recruits or people being studied in their 20s and 30s. And if you think of conditions like obesity, the rate of weight gain in absolute terms is highest, regrettably, when you're in your 20s, the rate of weight gain. Obviously, you keep gaining weight and obesity becomes more prevalent as you get older. But actually, the origins of the problem lie in this period here, all the transitions of life, and we're not studying it very well. So there are some gaps there, and when we think about how to fill them, we need to be informed by those and where the science needs to go. But we also need a fundamental change in how we view the investment in cohort studies. So in the past, they were regarded as let's put it, driven by individual researchers, largely for those researchers' benefit. And that is not a sustainable proposition. So increasingly, they've been thought of as being invested in, not just for those individuals, but for everybody. And, UK Biobank has really changed the game on that, absolutely amazingly. But MRC wanted to take a leaf out of ESRC's book, who did it really well, to think of investments in data as infrastructure, as capital. They're the capital investment that we need to do our science. And that has really helped unlock some major investments that have helped perpetuate funding in UK Biobank, but as I'll show you in a second, have also helped us fill some of the key gaps in the portfolio. So I said that we need to keep investing in cohorts to enable the study of people living through different phases of life and different periods of time. So you can't have a birth cohort that serves all needs because times are different. So ESRC are leading an investment in planning an early life cohort study, which has been in a feasibility phase and should go out into the major recruitment phase shortly. But this was funded by the Infrastructure Committee of UKRI. And that committee also funded a UKRI investment led by MRC in a major new adolescent health study. This is a new national infrastructure focused on critical biological and social developments that occur in childhood and adolescence. It's going to be a cohort study with a data platform accessible to all researchers, very much like UK Biobank. It aims to recruit and retain, and that is a real challenge, 100,000 children and adolescents from the age of 8 to 18, and study them for a period of 10 years. It's going to use an accelerated design, by which we mean that you recruit all age groups simultaneously and then follow them up over 10 years. So a group of 8-year-olds studied till they're 18, 18-year-olds studied till they're 28, et cetera, et cetera. And that allows the cohort to study important different transitions, because this is a very important phase of life. So the phase going from childhood into early puberty, through adolescence, and adolescence into early adulthood. So this is a space that is unique, and it's going to be filled by this cohort that will recruit through schools, but will be designed with young people and those schools. And it really aimed at understanding the key transitions and biological and social drivers of health in this critical age period, and with a view to informing policy interventions. It's got a deliberate policy of being inclusive, so it needs to not seek to be representative of the UK as a whole, but representative of the range of important subgroups in the UK, which is a real challenging proposition. So hopefully this major investment is £63 million will stimulate an exponential increase in research in this age group in the way I think the UK Biobank has done in older adults. And why study adolescence? Maybe it's obvious. It's a critical period of biological, cognitive, and social change in which health-related behaviors are established, and obviously has important implications for current and future health. It's understudied. Adolescents are underrepresented in our cohort profile. If you start at birth, and birth cohorts are fantastic and important, by the time you get into late childhood, adolescence, into early adulthood, you get cohort attrition. So that's why this cohort deliberately started a bit later, to try and expressly study that period. The other thing is, I think most of us recognize that the challenges to health and well-being for today's adolescents are unique. Not really much point looking at, adolescent experience and childhood experience in the 1960s or something and trying to generalize that to today. Need data from today to inform our thinking about today's challenges. And that's where we think this study is going to be extremely important for the UK. It needs strong leadership, and I'm very pleased publicly. This isn't been announced through the normal challenges because of elections, but Cathy Sudlow has agreed to take on the role of being the director of the UK Adolescent Health Study, and I think that's a terrific appointment. We're very excited and look forward to working with Cathy as she takes this forward. So one of the other things that the 2014 report looked at was how could we enhance the use of existing data, how could we share better, and how could we harmonize methods between longitudinal population studies. And it is important to look at a few of the things they said in 2014, because I think they were very forward-looking. So they suggested cohorts should use standardized, validated collection tools and platforms, and try to align with leaders in the field like UK Biobank, which really makes future cross-cohort comparisons much easier. I think there's an increasing trend to do that. The second recommendation is a bit more challenging. The suggestion was that we needed to have broad and enduring consent for linkage to routine data sources, so that it could, and that would be necessary for all prospective studies and sweeps, by which it meant repeat assessments of participants. I agree with that recommendation, but I think it's very challenging at a time when some of the bodies responsible for enabling or allowing linkage take a rather narrow view. If you don't mention things very specifically, that's problematic, and then it makes it difficult for cohorts to rely on informed consent as the legal vehicle for getting linkage, and many have to rely on Section 251 approval. And I think this issue needs resolution. If you talk to the participants in cohorts, and we led a participant panel on Monday with the Epic Norfolk cohort, that people do not understand if they've said, I've signed a consent form that says I want you to be able to link to my health record data. They do not understand why that is not possible. And we have had 10 years of talking about this, and it's not yet solved. And it may require political will. It may be that all the bodies are paralyzed, and it needs some sort of political will to break the inertia. So, and the last point was about skills development. And that's where Closer, which is an investment to enable development of methods and sharing of those methods with the community, where Closer has been a very important interdisciplinary partnership leading social and biomedical longitudinal population studies in the UK. So this has been an important investment and has made progress. But what about linkage? So most of the cohorts that were looked at, there were 32 back then, there are many more now, most of them had consent or thought they did. Fewer had consent to administrative data or the broad census and environment. And the MRC has invested with others in the UK longitudinal linkage collaboration, which is led by Andy Boyd. It was originally started in the period of the pandemic for reasons of trying to study that disease. And at that time, obviously, getting linked data was very possible because of political edicts and the public health salience of the problem. It's become harder subsequently. And I think the UK LLC, which is an investment from MRC, is an important thing to try and make some progress on the linkage that our participants really think is important. So what about the challenges of bringing smaller cohorts together? Well, the 2014 report said cohorts should ensure that they are discoverable. I think that goes without saying, and that's much easier now. And that the processes are put in place to ensure that people abide by data sharing policies. They need to be accessible and enable data sharing and access to samples. Let's see how that's happened. They need to adopt common data standards, sharing knowledge and improving metadata quality. I would argue limited progress on that over the last decade. And you need to enhance the use of digital technologies where possible, particularly in new phases of the study. So MRC and ESRC think that this is an area where we need to specifically invest, and we need to invest now, because there are 2.5 million people in those cohorts that I've mentioned here. Okay, Raghib's going to talk about OFH, which has 1 million, and we'll get 5, he promised me. But we have an obligation to try and use the data and the samples that those 2.5 million people have donated. And there is a time window in which it's useful. So MRC has established with ESRC a joint resource called Population Research UK, and it's led by Nick Timpson and Alyssa Goodman. And its vision is to maximize the use, innovation, and benefit from the UK's rich collection of these longitudinal cohort studies that I've been describing across the social and biomedical science fields. And in a way, bringing the cohorts together is important at a strategic level because it's easier to argue for big sums of money, sometimes with capital, for a big investment like the adolescent health study than it is for multiple small ones. But I think it's also important scientifically because we need to try and analyze those individual cohorts, which individually probably won't contribute much to pharmacogenetics or stratified medicine. But together, they have the potential to do that, but only if it's easy. So in the preparatory work for Population Research UK, HDR UK did a review about experiences of using LPS data from key stakeholders. And in green are the things that are kind of easy, and in red, the ones that are really difficult, and all colors in between. So it's pretty easy to, these days, to analyze single data sets. But for many things, particularly if you want to start stratifying, that's never going to be enough. So we need to bring data together. Finding cohorts, moderately difficult. Understanding what's in them, really difficult. Metadata, still pretty useless. Getting access to the data, red. It's too difficult. Linking the data, still red, not sufficiently easy. Analyzing things together, pretty hard. So I think for a while, people focused on findability. They talked about directories. That's really not the issue. The issue is how do we make it easy for people to get hold of the data or get access to the data in a streamlined way and analyze it efficiently. Because if it's difficult, people just won't bother. Why would you bother to try and meta-analyze across multiple cohorts if you could just go to UK Biobank or OFH? It's got to be as easy as that to do it across all the other cohorts. Otherwise, people will just vote with their feet. They won't do it. And they'll miss the opportunity of the massive amount of elapsed time and the massive amount of heterogeneity that there is in the existing cohort data. The other problem is, in a way, a nice problem. So there's been a massive investment in infrastructure across different sectors in trusted research environments. So this is a UKR investment in data and analytical research environments, UK. I think I'd be interested in your perspective, that I think what's going to be important in the future is if I, as a researcher, want to get access to any of these TREs, it'd be nice if the requirements to do it were unified and I could be authorized once, not 30 times. And if I'm trying to get to 30 cohorts, maybe it'd be easier if it was in one environment rather than 30 separate ones. But be interested in if we got time for discussion on that point. So here's a case in point. So MRC have invested in exome sequencing of 4 different cohorts. Born in Bradford, this has been done by in collaboration with the Sanger. Born in Bradford, ALSPAC, the Fenton study, and the Millennium cohort. So here's exome sequencing in these individual cohorts. But there isn't today a TRE that is the obvious place to go to, a singular TRE, where researchers can go and analyze these 4 data sets together, which I think is necessary and is an issue for today. So the final thing I wanted to highlight was, what about ongoing investment in existing cohorts? And I think the future lies in strengthening Population Research UK and trying to secure major investment for those cohorts as a singular entity, because that's the route to getting sustainable infrastructure long term. Now, in thinking about that, I think we have to take a radical look at that existing cohort landscape and treat cohorts differently. So the MRC have come up with a new life cycle of cohorts. So most cohorts go through an active phase where they're recruiting people or having face-to-face contact with them. That's fine. But there comes a point where, with cohort attrition, the active follow-up of participants and requirement for face-to-face contact with them becomes challenging, and you need to move to a passive phase where you don't have that direct collection of data from the participants because of the issues of attrition and selection bias, but you continue passive record linkage. And that's a very important phase, and many of the cohorts are in that now, and we have to maximize that information. But we need clarity about the routes to getting that. So MRC has a funding mechanism for cohort studies with a strategic oversight group that I chair to look at new cohorts and ongoing investments in existing ones in the light of this landscape review that I've just talked about. But one of the challenges is this is the duration of follow-up of some of those cohorts in 2013, 14, so 10 years ago. So the top one there is the Hertfordshire cohort. Now, many cohorts in Britain have now reaching a phase where many of the participants are very elderly. And you could argue that there's a real problem that even just continuing with passive record linkage, which has a cost, it's not a free gift, it has a cost. Even that is probably not worth doing anymore for some of these, because the incremental benefits of another 5 years of follow-up are very small. And the real problem in the UK is we haven't had a mechanism for saying, okay, how are we going to invest in these cohorts in a different way to make their data available and make the samples available when they cease that phase even of passive follow-up. So we are investigating and doing a pilot study funded by the Strategy Board at MRC of moving some existing cohorts to a data repository phase. So they would cease to be active cohorts with ongoing open ethics and would become research databases, which has different implications. And the sample use would be enabled continually, but they'd move from being active open studies to being research tissue banks, which has a different regulation under HRA. Now, this has not been done, and it's not been done because many of the cohorts haven't reached this phase. But my view is that many of them are about to, and unless we've got a funding mechanism for an exit strategy that enables the data to be continued to be used, that would be a waste, particularly for those participants who gave their samples and their time. If we just throw it away, if we rely on universities to do this, it's not going to work because people will retire and the samples and everything would just get lost. And I'll speak from experience. So I think we need to do this as an active new investment. So I hope I've given you the sense that there's a richness in the cohort landscape in the UK that we need to continue to capitalize. But we need a strategic framework for doing that. We shouldn't just keep investing in the existing landscape because it's there and we have a rich tradition. We should do it because there's value in the information. And I think, pharmacogenetics and stratified medicine is a key example of that. So with that, I will close and happy to take questions, Munir.
Thank you very much, Nick. Great talk. Comments or questions? Please, Bill.
Thank you. I was very interested in your comment just right at the end about the value of retaining information and following up individuals who are getting much older. Because I know that there are a number of studies around the world, particularly in China and other countries, where they've actively recruited individuals of 100 years and older because there's something biologically very interesting in people who have survived to such an age with perhaps not the spectrum of illnesses that you would expect associated with elderly individuals, and that might tell us something about some protective mechanisms that come into play.
If I gave the impression that I didn't think that was of value, Bill, I didn't mean to. So I think if that's an express desire, aspiration of a study to study that important age group for that reason, that's just fine. It's just, I think some cohorts go with the Magnus Magnuson, sort of style of wanting investment. I've started, so I'll keep going, and I'll keep going until everyone's dead. You have to have a slightly more active reason, and the reason you articulate is just fine, but people have to articulate it.
Sue.
Nick, that was a really fantastic talk. I just wanted to ask a question about feedback to participants in cohort studies, and particularly because you made the comment about access to longitudinal health data for cohort studies, but how can we get the benefits back to caring for these individuals instead of having to wait for scientific publications? That's one of the challenges we have with UK Biobank. We have to wait 5-7 years before we can see that evidence and we can act on it in the NHS. So should we be doing more? And I know probably Raghib's going to cover that too.
I think that's what's known as a leading question.
It is.
So should we be doing more? Yes. I think that older cohorts had consent arrangements that made that challenging.
Yeah.
Also had a view that they were, before we decide to do any feedback or et cetera, we had to build the evidence base. That was probably okay then, not sure now. I think some cohort leaders have a view that there's something pure about observation and we should never do anything because within a cohort because it'll taint that observational nature. I think that's a nonsense. I think more recent cohorts are building in consent for feedback. I happen to think that's a good idea. I think we should be doing more embedded trials within cohorts and expressly setting it up so we can do that. Otherwise, the pace of innovation and development is too slow. I agree with you.
You talked about TREs and having single TRE, for example, for those 4 studies and so on. That may be quite difficult to have single TREs, so people are talking about federation and so on. So I don't know whether that's, there are challenges there as well, so I don't know whether that's been tried and whether that's been discussed.
No, I mean, I agree with you, Munir. I think the idea that there's going to be a singular entity in the UK that we can all access, that's never going to happen. But if we go for federation or multiplicity, I think what I was trying to stress is I do think you've got to think of it from the user perspective. And some of these, you need, to get on it, you need to do this or that training and prove this or that. Somebody's got to sort out all that front end so it's simple for the poor user. Because I do fear that people just won't bother if it's difficult.
Great. Well, thank you very much, Nick. Thank you. Okay, let's move on to the last talk of this session. Great pleasure to introduce Professor Raghib Ali, who's Chief Executive and Chief Medical Officer for Our Future Health. Raghib has a long-standing history in leading initiatives like this, including he was involved in the UK Biobank, has been involved in the UAE Healthy Future Study, etc. So Raghib, keen to hear about what's going on with Our Future Health now. Even bigger initiative than everything else.
Thank you. Thanks, Munir, and thanks to the BPS for inviting me. I think it's just over 15 years since I last spoke at one of these meetings when I was working on a cancer research network in India, as you may recall. But today, as Munir said, I'm here to speak about Our Future Health, and it's already been mentioned a couple of times, and I hope by now all of you are somewhat familiar with it, at least have heard of it, and I'll go into a bit more detail about what we're trying to achieve through the program, the current status, and the opportunities for really many types of research. I've highlighted 3 opportunities there: population health, genomics, and prevention research. So just to kick off, although this may be familiar to some of us, we are facing huge public health challenges not just in the UK, but across all high and middle income countries in terms of increasing burden of common chronic diseases with an aging population. And some of the things, the trends that we've seen over the last 100 years in terms of increasing life expectancy, and particularly healthy life expectancy, have started to go into reverse. This is not just an issue for older people, but also working age populations. And there are also widening inequalities, sadly, between the most deprived and least deprived populations, and also by some ethnic groups. So, as I said, these are very big challenges, and research and studies like Our Future Health and all the other studies we've heard about this morning will have an important role in trying to understand better ways to change that picture, particularly focusing on prevention. As I mentioned, life expectancy, really after 100 years of going up, has started to decrease. This is partly driven by COVID, but the trends were there beforehand. Increasing levels of obesity, of course, are partly responsible for this. Also, certain severe mental health disorders, particularly in younger adults, have also led to this decrease. Increasing levels of multimorbidity as we age. So by the age of 50, most people have at least one common chronic condition, and certainly by the time people reach their 70s, the vast majority have more than one. So this is not inevitable, though. There are populations across the world where we see much better healthy life expectancy, but in the UK we have a particular challenge where we're not seeing that currently, and if anything, we're going backwards. In terms of working age populations, again, since the pandemic, we've seen this very large increase in people being off work due to ill health, particularly due to musculoskeletal disorders, mental health disorders, and long COVID. And also, you can see the list there. So this is a huge challenge. Being out of work, of course, is very bad for your health, but it's also very bad for the economy, and therefore bad for public services in the long term. As some of you know by background, I'm a consultant in acute medicine, so I spent most of my early career working in A&E, seeing patients at the end stage of a disease process, whether a heart attack or a stroke or kidney failure or a cancer that had already spread. And of course, most of us as doctors or working in acute medicine would be familiar with this. But this is not inevitable. It's not, these diseases develop over years, if not decades. And what we're trying to do, as one of the things we're trying to do through Our Future Health, is to change from this current model of care to a new approach, which I'll outline in the coming slides. Just briefly, there are different types of prevention, and we're focused on Our Future Health, particularly on primary and secondary prevention. I think these slides actually will be shared afterwards as well, so I'm happy for people to look at them afterwards, and we don't have time to go into detail on all of those. Just a brief point on, historically, for good reasons, we're focused more on moving the whole population to the left in terms of risk factors. So things like smoking, high blood pressure, salt intake, etc., physical activity. And that certainly has the biggest impact at a population level. But that can be complemented by, this is a very famous diagram initially from Jeffrey Rose many years ago, but it's tackling the right-hand side of the distribution and identifying those people who are at highest risk, and not necessarily treating them medically, but also through non-pharmacological interventions. Through recent advances in technologies, some of which are listed there, certainly not an exhaustive list, but polygenic risk scores, for example, using protein biomarkers, circulating tumor DNA we heard about earlier, imaging techniques, etc. There are newer ways that we can identify those people who are at highest risk, predict who will develop diseases much more accurately than we can currently, diagnose the disease earlier, and therefore intervene earlier. And that's the model that we're trying to move towards through Our Future Health, or to establish the evidence base for this approach. So there are different terms for it. I've used precision public, or others have used precision public health there as well, precision population health. It's an aspect of precision medicine or precision prevention, which, as I said, is about identifying people at high risk earlier, diagnosing disease as early as possible in the pre-symptomatic phase, and intervening with both pharmacological and non-pharmacological interventions. So Our Future Health, the idea for this actually came out of the life sciences strategy, I think in 2019. It was decided to fund a project which would focus particularly on early detection, early intervention and prevention of common chronic disease. It came out of the life sciences and industrial strategy deliberately as a partnership between government, industry, medical charities and the NHS. So it would advance both public health and the life sciences industry, which is a key source of innovation for the UK. We've been very fortunate to be able to build really an unprecedented collaboration with many partners that are listed there. Of course, the NHS is the key partner. They've been critical in our recruitment of participants through the NHS Digital Trial Service. Going forward, we're working closely with Sue and colleagues for future feedback of disease to participants in the coming years and decades. But for now, we're in the recruitment phase predominantly and providing access to researchers to our data. We're working, as you can see, with medical charities there, Life Heart, Cancer Research UK, British Heart Foundation have also joined recently. Many companies from both pharmaceutical industry and the diagnostics industry, and also many, many charities. And what we've achieved to date has really only been possible through this collaboration. And of course, most importantly, the public who have agreed to join the study in very large numbers, as you'll see. This just summarizes some of the main features of our Future Health. What most people are familiar with is the very large size, so the 5 million number, which, as Munir mentioned, the first study I worked on was UKPGx back in nearly 20 years ago now, when we were just starting. And even at that time, we thought 500,000 was maybe not possible. It was a very ambitious target, but of course, it was successfully achieved and has really led the way for cohort studies globally, particularly in terms of the access to data that they've provided and the number of publications. But it's not big enough to look at all common chronic diseases, or even certainly not rarer diseases. And many cancers, it's not really big enough to look at many cancers. And the age group that was chosen, 40 to 69, it was not really possible to look at the early stages of obesity or mental health disorders, asthma, etc. And also it wasn't, because for very good reasons at the time, it wasn't possible to really recruit large numbers from ethnic minorities or from more deprived populations. So there's a gap in terms of the type of people that have taken part in cohort studies before. I mean, Nick showed, there's a very big gap for younger adults, working age populations, but also ethnic minorities and people from more deprived backgrounds. And there are two other key differences I'll come back to in the next slide. The main point on this is that it's not just an observational cohort. It exists, our Future Health exists as a resource for discovery and translational research, and that's shown here. So that's what we're primarily trying to build, a resource for researchers, building on the model that UKPGx used, but not just for discovery research, not just an observational cohort, but a cohort that we can go back to our participants, give them feedback about their risk of disease, as is mentioned here, using both genetic and non-genetic information, and to enable assessment, for example, of risk-stratified screening programs, as opposed to now when we mainly just use age for most of our screening programs, but that misses people who are at higher risk at younger age groups, both based on genetic but also non-genetic risk factors. And crucially, the opportunity to go back to our participants for clinical trials, interventional studies, biomarker validation studies, etc. This has never really been done at scale before. And the key challenges for our Future Health, one was, of course, to recruit 5 million people in the first place, which we've made good progress on. But the second two aims here really are unprecedented in what we're trying to do, to work with the NHS across all 4 nations of the UK to deliver that feedback and to run these recontact studies. I will speak briefly a bit more about the genetic components of the study, because that's particularly relevant to the pharmacogenomics, which, of course, we're discussing at today's meeting. So we have a custom genotype array developed in collaboration with Illumina, with approximately 700,000 variants. It's been optimized for the UK population as a multi-ethnic population, which we are now, to look at factors that contribute to disease risk, particularly polygenic risk scores, combining that with other data of integrated risk scores, but also for predicting blood types and assessing pharmacogenetic variants. We're working closely with Genomics PLC, led by Peter Donnelly and Gil McVean, to provide those polygenic and integrated risk scores. This has a bit more detail about the custom genotype array, and again, I won't go through all the points there, but we've tried to make it as comprehensive as possible to maximize the utility of it for research purposes, and as I said, to build the evidence base for the various uses within the Our Future Health resource. One of our key aims is to work on establishing the utility, clinical utility and cost effectiveness for the use of genomics in population health and prevention. So for most common chronic diseases, existing risk scores don't include the genetic elements, so whether it's diabetes or cardiovascular disease, breast cancer, colorectal cancer, etc., where we have screening programs. But particularly for younger populations and for certain ethnic groups, it's likely that adding genetic information to existing risk scores will improve the utility of those programs. But we need to show that. We need to provide the evidence for that. We talk for hundreds of years about prevention being better than cure, but of course we need to be able to show that through rigorous and well-designed research studies to demonstrate both clinical utility and cost effectiveness, and also to assess psychological and behavioral aspects of giving that feedback. For common chronic diseases like diabetes and heart disease, it may be relatively straightforward. But for other diseases where we don't have necessarily good treatments or where the genetic component has higher penetrance, again, there's a lot more work to be done. And we look working closely with other cohorts, other studies like Genomics England, with the Genomics Medicine Service, to enable us to maximize the opportunities, as I said, for research. Just a couple of slides on pharmacogenomics. So we have, as I said, a number of, well, the points are listed there really as to how we can undertake research studies looking at particular alleles and variants across 50 genes amenable to many downstream tools. Those of you who work in this field will be more familiar with these terms, but you can see there that there are opportunities for research across a number of areas of pharmacogenomics. And the next slide lists some of the potential use cases as well, and particularly the opportunity to go back to participants, to do recontact studies, to look at not just efficacy of drugs, but also adverse reactions. And Munir has really led this work for decades now and has established already an initial evidence base for use cases that can be implemented across the NHS. But there are, of course, many, many more opportunities for that. And we hope that Our Future Health will provide the evidence base for that. So I won't go through all the points on here, but I noticed that my colleague Michael Cook, Executive Director of Science, Our Future Health, is also here and has really led this work for Our Future Health, who will be happy to answer questions during the break as well. I should stress that Our Future Health is not only a genetic study. We are looking at the whole spectrum of risk factors, including individual lifestyle factors, social determinants of health, because of course for most diseases, the genetic component is actually relatively small. And unless you have data on all these other risk factors, you won't be able to identify those people who are at higher risk as well. And particularly, as I said, people from ethnic minorities and more deprived backgrounds, and the whole geographical kind of representation of the UK, we've got that now for the first time in Our Future Health. For those of you who joined the study, you'll know that we have a fairly comprehensive baseline questionnaire, which tries to capture as much as possible about those risk factors. Of course, there's always more you can collect, and we will be going back to participants in the future to collect more data. But at baseline, we have fairly comprehensive phenotyping by questionnaire. Going forward, we will be able to collect a lot more data, and we've kept the baseline phenotyping relatively light because 1 for 5 million people, even any measurement costs a lot of money, but also because of time. To get people to take part in the first place, we're very conscious of how much time we can take from people at baseline. But having a digital cohort makes it much easier to go back to people, to get additional information from questionnaires, to enhance our exposure assessment, to do cognitive function testing, for example, collect data from wearables, whether physical activity data, dietary data, pollution data, etc. And also to link to additional databases. So Nick talked about linking to administrative databases. We're very keen to do that. We do have fairly broad-based consent for health-related data, but it will be necessary for some types of administrative data to go back to participants and reconsent them. But again, that is easier. All of our cohort have consented to online already, and it's very easy to go back to them, ask them to consent again. Even if half of them agree, that's still a very large number of people within a 5 million person cohort. And then looking further ahead, and of course this does depend on future funding, there will be the opportunity to use our biobank samples for whole genome sequencing, proteomics, metabolomics, methylation, etc. So that's the kind of looking ahead for the next 2 to 3 years is what the opportunities will be. So if you haven't taken part in Our Future Health, this is what it involves. You go to our website, you register online, consent after reading the information leaflet, fill in the questionnaire, and then book an appointment to give a blood sample and have some basic physical measurements taken. So it's height, weight, blood pressure, waist circumference, and a point of care test for cholesterol. It is a new study, as Nick said. We only launched in October 2022, so just over 20 months ago now. It was towards the end of October. We've been fortunate to have very helpful coverage in the media, but most of our participants actually come through receiving a letter through the NHS Digitrial service, and about 80% and about 20% have come through hearing about the study in other ways. We've been very cognizant of the fact that most cohort studies in the past have not been fully representative of the diversity of the UK population, and there are good reasons why people from more deprived backgrounds, from ethnic minorities, and certain parts of the country haven't been able to take part in the past, particularly rural areas and coastal areas. What we're trying to do through our Future Health is to enable everybody in the UK to take part. Of course, the first stage, anyone can take part because it's online, but also to provide clinics within a 5-mile radius of nearly everybody, 90% of the population. Of course, in rural areas, that's a bit more difficult. But we have worked also with groups from ethnic minorities, from more deprived populations, to design our materials to make them as user-friendly and understandable as possible. And we also provide reimbursement to participants to ensure that no one is excluded due to financial reasons. So on the left of the screen there, you can see we've already had about 200 venues. We will eventually have about 400 venues across the whole of the UK. This shows really an unprecedented rate of uptake for not just in the UK, really anywhere in the world in terms of people taking part. And this is a great advantage of doing studies in the UK. We have a population which are willing to take part, and with the NHS, of course, the unique opportunity to track people prospectively and also to get data retrospectively. So you can see in terms of the numbers, already 1.6 million people have consented. That number will always be bigger than the other numbers because you can consent anywhere in the UK. Then people have to fill in the questionnaire and give a blood sample. You can see we're approaching, well, over 1 million people with questionnaires and approaching 900,000 people with blood samples. Actually, this data's a week old now because it's from last week. So we have about nearly 900,000 people now with blood samples. And the full participants are those who have completed every step, and that's about 830,000 as of today. So every week we see around 15,000 people. Every month around 60,000 people become full participants in the study. This is a comparison to other programs, not just in the UK, but globally, particularly studies in the US and China, for example. We have found a methodology which has enabled us to recruit just much faster. This is people who are full participants, much faster than previous studies. This is a comparison to the All of Us study in the US, which has quite similar aims to our Future Health. They're aiming to recruit 1 million people. They're about halfway, they're just over halfway. They have over 500,000 full participants and about 800,000 consented participants. As you can see, as I said, we found a way not just to recruit much faster than other studies, but also at much lower cost, about less than 1/10 of the cost, for example, of the All of Us study. In parallel, although we spent much of the last 3 years trying to optimize our recruitment journey and to maximize the numbers who join the study, we've also wanted to make sure that people can access the resources as quickly as possible. So researchers from academia, from the NHS, from industry can access the resources as quickly as possible. And this is also unprecedented. So as of now, you can already access the resource. You can apply to our access board, which is independent of our Future Health. It's chaired by Nick, and so you can apply now. We've managed to do that much faster than UK Biobank, of course, was a pioneer, and we've been able to learn from their experience. But we've been able to do it much faster than UK studies and also, as I said, the American equivalent. This is a map that's a little bit out of date now. As you can see, it's from March. But even in March, we had participants from every part of the country already, from every local authority. It doesn't project very clearly, but there's even someone there from the Orkney Islands. So we really have managed to get the whole spectrum of representation. This is really important because previously, even UK Biobank, only 40% of the population was able to take part in it because they were those that were invited. And it was only, as I said, in the age group 40 to 69. I mentioned we have tried to maximize uptake from underrepresented groups. And this is the demographics of the first roughly 1.5 million participants who have consented. And it's very difficult to get a fully representative sample. We knew that younger people, more deprived populations, and ethnic minorities would be less likely to take part. Ethnic minorities in particular some of them are much younger. So Pakistanis and Bangladeshis are much younger than whites, and that's one of the reasons they take part in lesser numbers. But even given that, in terms of absolute numbers, we have by far the largest cohort, I think, in the world now of 18 to 40-year-olds, and in the UK of most deprived quintile and of non-white ethnic groups. That's for those who have consented. Even if you look at those who have given blood and completed the questionnaire, very large absolute numbers, although I mean the percentages are not quite, as I say, representative, but certainly we've done better than previous studies. But we want to do even better. We want to Our aim is to have 10% of the cohort from the most deprived quintile, to 500,000, 10% from non-white ethnicities, again 500,000, and 10% from the 18 to 40 age group. So that will really transform our understanding of the disease in these three groups. This just provides a breakdown by individual ethnic group. Again, this was somewhat predictable from previous studies and because of the age profile of these individual ethnic groups. Interestingly, our highest uptake is actually amongst the British Chinese community, so even higher than British whites, and the lowest is amongst British Pakistani and British Bangladeshis, and that's mainly due to their much younger age profile. But also there are issues of trust, and we saw this during the pandemic. I mean, some of the work that I did in trying to improve uptake amongst those groups, there is a lack of trust, often for good reasons, but amongst the Pakistani and Black African, Black Caribbean populations, and we need to do more work to build that trust, that people feel confident to take part in research studies like Our Future Health. Our cohort, I'm not going to go through all of the baseline characteristics, although some of it's available on our researcher website. In general, we're not finding that our cohort is much healthier than average. It is slightly healthier, which is kind of inevitable, but not much healthier. So if you look at BMI, smoking status, cholesterol, or blood pressure, we have very high prevalence of existing disease, not far away from the distribution you see in the representative health survey for England. And that's good because we don't want to have a very healthy cohort, and we want to have a cohort that's representative of the population as a whole. I've just highlighted on this slide some of the key and unique aspects of Our Future Health. It is a translational cohort, as I've said. The opportunity to do recontact studies, recall by phenotype, recall by genotype, will really transform the landscape for clinical trials in the UK and catalyse particularly trials in prevention and in younger age groups that haven't really been done before. I've mentioned most of the other points already on this slide, but it does highlight the opportunity to look at diseases in younger age groups, so obesity, asthma, mental health disorders, addiction, but also diseases of aging, particularly dementia and Alzheimer's. At the beginning, I showed how large numbers of people are off work due to ill health, and I said, of course, that's bad for their health, but also bad for the economy. Again, there's an opportunity through Our Future Health to understand better why people are off work and also what interventions can be effective in getting people back to work. And that, of course, will help to improve their health. So this is an opportunity for us, which really hasn't been possible before. Because it's such a large database, there will be very big opportunities for those who work in AI and machine learning to use the resource, combining the questionnaire, physical measurement, NHS link data and genetic data. So yeah, again, very big opportunities for research using AI and machine learning. The final part of my presentation is on opportunities to access the data. So we have a trusted research environment already established, which, as I said, you can apply to access now. There will be opportunity for trusted partners to also apply for their own TRE, and that will be a process that will have the same criteria as ours. And also, as you heard about the NHS secure data environments, opportunities to work with those as well. You can apply now when you go to our website. So the researcher website, as I said, is live. Actually, as of today, the data available will increase significantly. So there'll be data on about 1 million questionnaires, about over 300,000 genotypes, and over 600,000 linked NHS data. This is the first release which is going to include the NHS linked data. And this is really why the UK, in many ways, does lead the world in this type of research. It's because of the opportunity to link to the largest single health data set in the world, which there are some challenges, as we heard about earlier, particularly with primary care data, but they're hopefully close to being resolved. And it certainly complements the data that we're collecting and will make the resource really uniquely valuable for prospective research and interventional studies. As I said, these slides will hopefully be shared after, so if you're interested in some of these numbers, you can look later. But we have already quite a few researchers from academia, from the NHS and industry that have applied. Looking ahead a little bit, so by the end of this year, we'll start to provide imputed data from the genotype array, physical measurements data, and new data that I've spoken about that will become on stream in 2025 and 2026. By this time next year, it will be a really huge resource. I mean, by far the biggest resource of its type globally, accessible to all researchers globally. So the opportunities really are unprecedented. This is my final slide. I mean, this is what we're trying to achieve through Our Future Health is extremely ambitious. We've made a very good start. We have a long way to go, and I hope, working with many of you in the audience, that we'll be able to work together to achieve those aims. Thank you.
very much. Any questions for Raghib? Can I just ask about the link data?
Sure.
Nick talked about the difficulties in that. So I don't know how far you got. Just expand on what you're doing with the linked data sets and how whether there are going to be difficulties.
Yeah, so for those of you who are not familiar, at the moment and for many years, it's been relatively easy to access secondary care data, so hospital statistics, cancer registry data, death data, and there are other data sets which we can access as well, but they're all from secondary care. The primary care data was available during the pandemic, and I mean, some of my own research used it to identify why people in ethnic minorities were at higher risk. So we know it's technically possible, but there are information governance issues, particularly that need to be worked through with GPs, with the Royal College of General Practitioners and the BMA in particular, that have been going on for years, but there has been progress made. Actually, it's not the same across the country either. So in Scotland, for example, it's already available in Wales. It's in England, really, where we just need to get through that last stage. But we have made good progress. I mean, I'm relatively new to these discussions, but from what I've seen over the last year, I think we can be more confident that by this time next year, it will be available.
Okay, great, thank you. Sorry, who have got there first? Yes, please go ahead first, and then one over here, Arun Hingorani here.
Thank you. Margarita Perez-Casal, Liverpool Health Partners. Really, really valuable tool, fantastic thing, and I'm one of those who is sign up. But one of the things that I can't help but always kind of like being slightly skeptical about is, are we actually capturing the worried well? And what are we doing for all those that, you know, and I'm coming from being in Liverpool, there is a massive population which is underrepresented possibly. What do we do to capture that? Thank you.
So that is always a challenge in these types of cuts. We certainly saw it in UK Biobank, where the cohort was much healthier than average. I showed some of the data points we have already in terms of prevalent disease. There's a lot more data we have. I mean, it's not that it's completely the same as you'd expect from the general population, but it's not far off, and it's certainly closer to the population average than UK Biobank. So we have, I mean, the things that we've mainly done to try to improve uptake from areas that have historically been underrepresented is to keep our clinics open in areas like Liverpool, but also all the major cities, but also coastal areas where we have huge inequalities, is to keep those clinics open for much longer. So in the areas where people tend to be healthier and less deprived, the clinic might only be there for 4 to 6 weeks. In areas like Bradford, Liverpool, Manchester, London, we have clinics open for years. Like literally, they've been open, some of them for nearly, over 18 months already. So that's just to make it as easy as possible. Providing reimbursement has also been helpful. We saw that improved uptake amongst more deprived populations, ethnic minorities, and younger people. So it's definitely a concern, but one that we're very committed to addressing.
Okay, last question.
Thanks. Amazing progress. Question about if you can say something about follow-up of positive results and the approach to that.
Do you mean genetic or all?
Anything that comes out. How do you,
I mean, to Sue's question earlier, this is one of the big challenges, of course, how to do this, whether it's genetic information or non-genetic information. And I think Biobank, for good reasons, 20 years ago decided it was not something they should do at that time. But now we do have the opportunity to see how that can be done at scale through our Future Health. I think for certain diseases, it's easier. So if you take diabetes as an example, or cardiovascular disease, where we have an existing, at least in England, an existing program through the NHS Health Check, we can provide people with information about their risk of disease, and they can access an existing service. For breast cancer, colorectal cancer, again, there are existing services. For newer approach, or for other diseases where we don't have prostate cancer, for example, or dementia, this is much more challenging. We don't have an existing service, and we'll have to work closely with the NHS to work through those issues. I mean, we have a number of advisory boards, an ethics advisory board, scientific advisory board, public advisory board, that we're also working with to get the views of relevant stakeholders. Over the last few months, we've also run what are called deliberative dialogues with members of the public and our participants to work through many of the issues on feedback and recontact, particularly the trickier ethical issues on what you feedback in terms of incidental findings, additional findings. So if I'm invited again next year, I'll be happy to update you, but we're in the early stages of that work.
Okay, great. Thank you very much.
Thank you.
So a big thank you to our 3 speakers. It's time for coffee now, so coffee's upstairs again. So if you come back here for 11:10, if that's okay. Thank you.
Welcome back, everybody. Thank you for coming back. I hope you are refreshed after the break. I wanted to get us going because we're running slightly over time, and we want to have plenty of time to hear from our speakers today, in time to get you for lunch. So I'm Vicky Chaplin. I'm the Senior Clinical Lead in the Genomics Unit at NHS England, and I'm really happy to be chairing this session today. This is the early career session, but I think you will agree, and probably even more so after hearing the talks, that we are going to hear from some really, really great speakers working on some quite pioneering areas that are probably going to inform the way that we are working in pharmacogenomics and stratified medicine now and much further into the future. So really excited to hear these talks today. The format is going to be, because we're running slightly behind time, we'll say approximately 14 minutes to talk and maybe about 3 minutes for questions after each one. So please do think about any questions you have. We'll stop after each speaker just for you to get those questions in. So without much further ado, I'm going to introduce the first speaker. So we have Dr. Emma Magavern. And she's going to talk to us about pharmacogenomics in cardiovascular medicine, insights from British South Asian cohort. And Emma is a clinical pharmacology registrar in London and a PhD candidate at the William Harvey Research Institute, QMUL, working with Professor Mark Caulfield.
Thank you so much, Vicky. We'll all have to try to live up to that introduction. And thanks so much for inviting me to speak to you today about some of the work that I've been doing. So pharmacogenomics, how variation in DNA can explain some of the inter-individual variability that we know exists in terms of response to medications. Now we know we can think about that in terms of risk of adverse drug reactions or risk of inefficacy. And when we break that down, we can talk about two pathways, so either pharmacokinetics, what the body does to the drug, or pharmacodynamics, what the drug does to the body. I'm going to hone in today really on the pharmacokinetic pathway, specifically metabolism, so how variations in DNA impact on metabolism of medicines to impact that variability and risk of adverse drug reaction or efficacy via those pharmacokinetic pathways. And really critical to that concept is talking about CYP450 enzymes, which metabolize most of the medications we prescribe and take. Those enzymes are encoded by genes of the same name. Simply put, we get one copy from mum, one copy from dad. And if you get two copies, one from each, that has a loss of function allele, you'll be a poor metabolizer of that drug, metabolized by that enzyme, and break that medicine down very badly indeed. One which works and one which doesn't, somewhere in the middle, or an intermediate metabolizer, and two that are working well, a normal or extensive metabolizer. So how significant is the problem of adverse drug reactions and that variability in response to medications? So I think epidemiologic population level data, which we've heard quite a lot about already today, says important and only getting more important. So we're prescribing a lot more medicines, 50% more between the last two census points in terms of NHS prescribing. And people are taking more medicine as they're living more years with multiple morbidities. The cost of adverse drug reactions are very high to health systems, and this represents a big burden in terms of patients and quality of life. So I think it's important. Now, pharmacogenomics is not the answer to all of these adverse drug reactions. It's not going to take that cost to patients and the healthcare system to zero. But it's one component, and it's one that we're not really actioning at the moment, which is why there's some room for improvement. Now, I've always wondered, in the scale of national level adverse drug reporting throughout pharmacovigilance, really what is the count and the percentage of ADRs associated with those pharmacogenomic drugs, those limited drugs where we have evidence for a drug-gene pair that would inform prospective prescribing, and where there's prospective randomized control evidence that using pharmacogenomics can mitigate that risk of adverse drug reaction, just a small subset of drugs, really. We couldn't find that answer anywhere in the literature, so we decided to have a look. So we analyzed yellow card adverse drug reporting data since the inception of the pharmacovigilance system in the 1960s, following the thalidomide tragedy, to ask this question. And what we saw was that in these yellow card reports, there were over 1,345,000 ADRs reported. And when we paired those genes and drugs together, so those pharmacogenomic drugs where there is that prospective evidence that we could potentially mitigate adverse drug reactions, those drugs were representing 9% of that overall composition of the adverse drug reports. And 3 in every 4 of those drug reports were linked with only 3 really key pharmacogenomic, pharmacokinetic pharmacogenes, and that is CYP2D6, CYP2C19, and SLC01B1. Now, we broke it down by specialty to say, okay, where could we make big impact in terms of adverse drug reporting based on this national perspective? And the big winner was psychiatry, representing almost 50% of those ADRs, but cardiovascular was the second at 24%. And if we then say, okay, well, how do those ADR reports for those pharmacogenomics actionable drugs overlap or correspond with prescribing prevalence? So which of those drugs are being overrepresented in ADR reports compared with how commonly they're prescribed? 70% of these medicines, which are coming up above that red line, are psychiatry drugs, again, showing big impact for that area. But in terms of cardiovascular, simvastatin is right up there as well. So how likely are any of us to have one of these pharmacogenomic variants that will mean that we could change prescribing if we had the information? It turns out from epidemiologic genetic data that that's essentially all of us. So then the question is, how likely is anyone to be exposed to one of those medicines? And this data from Professor Emma Baker's group at St. George's is based on English longitudinal prescribing data. And it says the answer to that is actually quite likely. So almost 90% of people by the age of 70 will have been exposed to at least one of those drugs where there would be an indication to change prescribing based on genomics. And by the age of 70, a quarter of those people had been prescribed 5. So we think personalized prescribing can do better in terms of a more tailored and less trial and error approach, helping us with efficacy, decreasing toxicity, and potentially having other beneficial effects. But there's a big catch here, which is that we want to make sure that anything we're doing is helping to promote health equality and doesn't risk inadvertently worsening that health inequality that we know is so sadly prevalent. And there's a problem in pharmacogenomics in that we know that in both therapeutics trials and genomic data, we're extrapolating from a pretty homogeneous, largely European ancestry population. So I really wanted to work with our local clinical population in my research, and I had the wonderful opportunity to work with a fantastic Genes in Health study led by Professor David Van Heel. At the time of analysis, they had about 44,000 participants with linked data, but now they've recruited more than 60,000, and they will increase that to 100,000. The participants in this study are of self-declared Bangladeshi or Pakistani ancestry, and this is a really important ethnic cohort for pharmacogenomics and implementation in the UK. We've just heard from the team at Our Future Health that British Bangladeshis and British Pakistanis are underrepresented, and that there's these issues that we know about in terms of trust. And it's also a growing percentage of our population, so 5 to 10% South Asian ancestry in the national census between the last 2 points. And this is a cohort of people where ancestry overlaps with deprivation within central and north-east London, and these participants are sadly, we know, 4 times more likely than European ancestry compatriots to suffer from cardiometabolic disease. So I've told you that this is a cohort that's not represented in medication licensing and therapeutic trial data, but don't take my word for it. This is sort of a representative example. Clopidogrel, here are the studies underpinning the licensure from the medicines regulator. And despite representing a quarter of the global population, there's no South Asian representation in any of these studies. So clopidogrel, as you know, is an antiplatelet agent, a P2Y12 inhibitor, and widely used in the secondary prevention of myocardial infarction, among other indications. It is a prodrug. Back to that pharmacokinetic pathway, it must be activated in the body by a CYP2C19 enzyme. Now, there's a bit of a catch here because loss of function variants in the gene that encode that enzyme are incredibly common. And there's been lots of orthogonal levels of evidence over the past 20 years showing that those loss of function alleles in that gene are then associated with high on-treatment platelet reactivity in people who take clopidogrel and an increased risk of secondary ischemic events. So in those people that have the genetic variant, that means they don't make the functioning enzyme, they can't activate the drug in their body, and then they don't get as much benefit from it. I said it's very common, these variants in this gene, and they are, but they're more common in some ancestry groups than in others. So in European ancestry, populations where this is most studied, about 1 in every 3 people have at least one loss of function variant in that gene. So those poor metabolizers, people carrying those loss of function genes, can't form the active metabolite and therefore derive less benefit from the medicine used as secondary prevention in myocardial infarction. But analysis was lacking, linking the genetic data with prescribing data and health level outcomes in a South Asian ancestry population because of that underrepresentation. So we performed a validation study to assess the prevalence of the CYP2C19 loss of function genotypes in this British South Asian ancestry cohort, and then to correlate those metabolizer phenotypes as predicted by their genetics with recurrent myocardial infarction in those participants taking clopidogrel. So first question, how common are these loss of function alleles? Second question, is that impacting on real world events from this real world health data? So here's the prevalence of predicted CYP2C19 phenotypes. 44% are intermediate metabolizers with one loss of function allele in that key gene, and 13% are poor metabolizers, meaning they've got a loss of function allele in that gene from mom and from dad on both sides. And that means that nearly 2 in every 3 people in this participant cohort would be expected to derive a decreased benefit from clopidogrel because of that predicted metabolizer phenotype. Now, how does that stack up with data that we're basing our decisions on for sort of population-level health economic studies and pharmacogenomics implementation? So here's our genes and health cohort. Here's European ancestry, and the trials that have been done, the prevalence of these loss of function alleles are almost the same as the European ancestry populations. So we're talking about 2% predicted poor metabolizers in European ancestry as compared with 13% in our cohort, so a 5-fold increase of people who, in a dose-dependent fashion, really can't drive that benefit from clopidogrel, and nearly 2 in every 3 with at least one of those loss of function alleles in this genes and health cohort as compared with, we said, 1 in every 3 within European ancestry populations. And then that next question, well, do we see a relationship in this very small cohort in terms of primary and secondary events? They're quite young, there's not been that many events to date. But yes, even in that cohort with only 39 recurrent myocardial infarctions, we can already see a statistically significant relationship between genetically predicted poor metabolizers who have been prescribed clopidogrel and a recurrent risk of myocardial infarction. Now, if we stratify that by metabolizer type, we've got a gradient here from poor metabolizers, intermediate metabolizers, normal, and rapid. So this is a gradient of participants who, based on their genetics, are better as you move right at activating clopidogrel. And you can see in these unadjusted percentage, this is just percentage of recurrent myocardial infarction, that you get a stepwise decrease in risk of secondary events of these participants with an acute myocardial infarction prescribed clopidogrel. So yes, the genotype does predict the medication response. So in summary, this UK South Asian ancestry population has a high prevalence of these loss of function alleles in CYP2C19, and that's known to lead to poor activation of clopidogrel, and is linked in this real-world health data with increased risk of recurrent myocardial infarction in those participants who had an acute MI and were treated with clopidogrel. But pharmacogenomics is not just about efficacy. We want to talk also about adverse drug reactions. And I showed you before that overrepresentation of reported ADRs associated with simvastatin from that yellow card reporting over half a century of data. And so I want to talk briefly about a study we've done with statins in that same cohort. As you know, statins are HMG-CoA reductase inhibitors, which are really first-line therapy, not only in the treatment of lipid disorders, but in the primary and secondary prevention of cardiovascular disease. So they're prescribed to huge swaths of the population, and this has resulted in a lot of concerns about potential adverse drug reactions. Patients are very concerned because lots of them take it, and prescribers are really concerned because it's highly effective, and we want adherence, basically, if it's reasonable. So the SLC01B1 gene encodes a hepatic transport protein responsible for the uptake and then subsequent clearance of statins. There's a well-characterized polymorphism, which is the STAR5 polymorphism, and that codes a missense variant, which reduces the function of that protein and therefore leads to increased exposure to statins. So we've got a genetic proxy for increased exposure to statins. And that is agent dependent to some degree. So you can see that the highest area under the curve for these STAR5 homozygotes are in these top 4 medications: simvastatin, lovastatin, pitavastatin, and atorvastatin. So we wanted to do a proof of concept study. We've got these amazing observational data sets. And linkage with medications is a barrier. We're very lucky in Genes and Health to have complete linkage with primary care data via CCGs for 86% of participants. But one of the questions is, how else can we use genomic tools to draw orthogonal lines of evidence from observational cohorts about a drug and a purported adverse drug reaction? So Mendelian randomization has been incredibly effective at doing this, but we thought, hey, can we use validated pharmacokinetic pharmacogenes to see something about a dose-response relationship which might tell us about purported ADR? So the literature was very conflicted about association between statin use and cataracts. Some studies claiming that there was an independent association with increased risk of cataracts in statin users. Some that actually the association was inverse, with a decreased risk of cataracts. And some that actually, if you adjust appropriately for confounders such as diabetes, there's no independent association at all. So we wanted to investigate this using that STAR5 allele of the SLC01B1 gene as that proxy for that increased exposure that you've seen in the last slide to statins. So these were the statins prescribed in primary care to our Genes and Health cohort participants. And you can see that it's primarily atorvastatin and simvastatin, which is important because those were in those top 4 drugs of a much higher area under the curve for those STAR5 carriers and homozygotes. These are the percentage of participants prescribed multiple statins, so most had tried 1 or 2, and there was no relationship between having tried more than 1 statin and that STAR5 genotype. So 35% of the participants had been prescribed a statin, and that, of course, comprises a high cardiometabolic risk cohort, which we know also will, by definition, have a high risk of cataracts. 1 in every 12 of those participants had one of those STAR5 alleles, which is our genetic proxy for increased exposure to statins. And only in the on-drug cohort and not in the cohort not exposed to statin, there was a significant relationship where there was a 1/3 reduction in non-senile young-onset cataracts in that high-risk cohort associated with the STAR5 allele. So those people that genetically had a much higher exposure to statins had actually a significant reduction in the rate of non-senile cataracts. So this suggests that a genotype stratified approach can be pretty helpful potentially to pharmacovigilance to elucidate some of those relationships. Take-home messages overall, though, really are the importance of studies like Genes and Health and other work that I know all the colleagues here are doing today, and we've heard so much about. It's really critical that we do better with engagement, that we build trust, that we get proportionate representation of our global populations in our research cohorts and therapeutic trials, and that we look at the broader socioeconomic, cultural, and political context so that we can leverage genomic tools to really target those health inequalities that we know exist in therapeutic response rather than inadvertently worsening them. So the example, for example, of intersectionality with that South Asian cohort in clopidogrel, you've got a population with high rates of deprivation that have very high risk of cardiovascular disease due to their background and that deprivation, who also have very high risk then of not benefiting from clopidogrel due to genetic resistance. So an intersection of risk there. A big thank you to everyone who has worked on hard on the research and the images presented here, and my funders. And sorry if I've run over, Vicky. Thank you.
I think we have time for one question. Does anybody have a question they want to ask? I've just got. Is there anyone with a microphone? Okay, thank you.
I'm sorry, it's all the way up. Hi, a great story. Ron Verschijk from Erasmus University in Rotterdam. Great story. I like the story also about the clopidogrel. I think the interesting part there is if there are variants in the 2C19 gene, which we do not regularly test for, so not the STAR2, STAR3. So did you investigate that? Are there variants that may explain, or looking at the myocardial infarctions in these other populations and then identify additional variants which we should investigate?
Thanks, Ron, and nice to see you. So this was done using the array data. So I focused only on 3 really well-validated variants, the STAR2, STAR3, and STAR17. There is now exome data. It's been available to me for about 4 weeks. And work is ongoing in that area because now we can characterize those novel rare variants. I think our power is probably going to be a bit low in terms of events right now to characterize health level outcomes in terms of recurrent heart events. But this is a longitudinal cohort. Average age now is about 41. It's a very young cohort, even if quite a polymorbid cohort. So that is absolutely the next step, incredibly important. And we know from Altman's work with UK Biobank that rare variants in these pharmacogenes are enhanced within those populations. So absolutely, super important, great point, and we're on it.
Looking forward to those results. Thank you.
Thank you.
Okay. Yeah, brilliant. Thank you very much, Emma.
Thank you.
So yes, something else to look forward to now for the next piece of work. But if we move on now to the next speaker, so we've got Dr. John McDermott. John is a clinical geneticist and an NIHR doctoral research fellow at the University of Manchester, and he's going to talk to us about developing an interoperable and scalable platform for the implementation of pharmacogenomics in the NHS.
Thanks very much, Vicky. Thank you. So, picking up from where Emma left off in her lovely presentation, we know that pharmacogenetic testing can be useful to avoid adverse drug reactions, improve medicine's effectiveness. But the question that we focus on increasingly within our group is how do we actually get that data into the hands of clinicians in a clinically relevant format and a clinically relevant time frame? It's all good and well as doing the testing in a laboratory, but unless you can get it into the hands of the people who need it, it has relatively little value. And the first step that we need to take is to take a step back and disaggregate the genetic testing approach from the model of service delivery. And these are often conflated in the pharmacogenetic literature. So genetic testing, put very simply, in pharmacogenetics, we can do single gene testing, where you're looking at single variants in a particular pharmacogene, let's say CYP2C19, or you can do panel-based testing, where you're looking at lots of genes at the same time. And there's different technologies you can use for those different approaches. But broadly, these are the 2 testing strategies that you can use. But the model of service delivery is distinct. So you can do reactive testing, you can do reactive with planned reuse, where you decide you're going to use that data again in the future, or fully preemptive. And just to explain what that means schematically, so this is what we do at the moment for most pharmacogenetic testing in the NHS. A patient presents, let's say they're receiving chemotherapy agents, you think they need DPYD testing, you take a blood sample, you get the results, hopefully weeks or months later. You can then use that data to inform the prescription. That's what we do at the moment. That's what many health systems around the world do. But there is a move towards reusing that data again over time. So reactive with planned reuse is where you still do the testing after a particular clinical presentation or a particular medicine is prescribed. You do the test, but then you embed that data within a patient's healthcare records so it can be used again in the future. And then you've got a fully preemptive approach, which some biobank studies around the world are exploring, where you don't wait for the patient to present. You do the pharmacogenetic testing at a set point in time, and then you have that data, ideally in their health records, whenever the patient presents again in the future. And there is a fourth approach that you could use that isn't the focus of this talk, but in theory, if you could develop rapid testing approaches, and we've done a lot of work working with GeneDrive in Manchester, taking a cheek swab, putting it into the machine, and you get a result within minutes. So that's a fourth testing approach, not the focus of this particular presentation. But what I want to stress is that you can combine these testing approaches with the different models of service delivery in different ways. And this is taken from a review that we're writing at the moment. And essentially, this up here is what we're doing currently in the NHS. Reactive model of service delivery with single gene testing. There is a move in the NHS and globally towards this quadrant, if you will, of the table, where you are reusing that data again over time. But that is an informatic challenge. It's complex to embed this intervention within a complex healthcare ecosystem. And reusing that data, rather than just using it in a very binary way, as displayed here, adds considerable complexity, both on an individual and a population level. So if I'm doing CYP2C19 testing to guide antiplatelet therapy around a particular presentation of stroke, let's say, that's a relatively binary interaction. But as soon as you're doing, let's say, panel-based testing, you've got multiple genes, and then that data is theoretically available for a myriad of healthcare presentations throughout that patient's life. And they're not just presenting with different presentations to the same healthcare setting, but they're presenting in pharmacy, secondary care, GP. How do you make that data available across that healthcare system? So it's considerable, it's complex for that individual. How do they make sure they can make full use of the data over the course of their life, but also on a population level as well? What we're talking about is distinct from rare disease and cancer predisposition syndromes. This is a genetic intervention potentially at scale. And one way to emphasize that is an interim analysis of a study that we've recently undertaken in Manchester called the IPTIP trial. This is the first 500 patients recruited to that study. And essentially, we recruited patients into a secondary care environment coming through our acute medical unit and our vascular wards. We did pharmacogenetic testing and then linked that to their historical and contemporaneous prescribing data. And what you can see here is that, as Emma has articulated already, the vast majority of these patients carry pharmacogenetic variants of interest. And so anything that's not green there is your non-wild type result. And we see, as Emma's already described, significant differences in distribution in pharmacogenetic variation between ethnicities. And then when you cross that with the prescribing data, and this is just from a single admission, this is not yet looking at their historical prescribing data, what we see is that around 80% of patients during that admission were exposed to a medicine for which pharmacogenetic guidance existed. Around 50% had results related to medicines they were exposed to in that admission, and just under 1 in 7 individuals would have had a change to that medicine that was considered clinically actionable as per the CPIC guidelines, and would have had a change to the medicine if it was available. So this is an intervention which would have implications for a huge amount of healthcare interactions on a day-to-day basis for the vast majority of the population. So complexity on the individual and a complexity on a population level. And just the final point to stress the potential relevance of this, we know that these medicines are incredibly common, medicines like antidepressants, antiplatelets, pain relief. The cost of treating conditions related to these medicines are incredibly high. So even small improvements in the effectiveness or safety of these medicines could have a major impact on a population level. And so how do we actually get this data so it can be used in clinical practice? How do we make sure that this intervention works within the NHS? And so the NHS England Network of Excellence for Pharmacogenomics and Medicines Optimization was established around a year or so ago now, and there's lots of different pieces of activity that goes on within that organization, but we focused on 3 specific areas: the testing approach, the data, and the actual service, how we get that out to people. And today I just want to focus specifically on that data element, because I think this is the key challenge in pharmacogenetics. The testing technology, we have a great genomic medicine service. I trust that they will be able to scale up their infrastructure and test for those variants, but getting that out into the hands of clinicians, from my perspective, is the key challenge. So first of all, the nomenclature. So as we've already described, variants in CYP2C19 can influence the effectiveness of clopidogrel. This is what the raw data looks like. If you're a clinical geneticist or you request genetic test reports regularly, you might be used to seeing nomenclature data like this. This means nothing to the vast majority of the healthcare workforce. And so if you present a PDF, a typical test report that looks like this, it's not going to be used in clinical practice. Fortunately, in pharmacogenetics, we have a nomenclature, star alleles, and that can be converted to your, used to inform or predict your metabolizer state. So what these variants mean is that you are a poor metabolizer for CYP2C19, and that might start to mean something to some clinicians, but still most people won't know what it actually means in practice. Actually, what it means is prescribe an alternative antiplatelet. And all of the stakeholder work that we've done, we've conducted a large number of focus groups, large number of interviews. That's what the vast majority of clinicians want to see. And so we've developed a fairly simple informatic tool to take that raw data outputs from different systems and automatically convert that into meaningful and structured data. And that's based on rules from the Clinical Pharmacogenetics Implementation Consortium. So that's a fairly simple problem, which we and lots of other healthcare systems around the world have started to address. But that real challenge, as I addressed right at the beginning, is how do you get that genetic variation out into general practice, out into pharmacies? And it's not just the different geographical locations, but everyone's using different electronic healthcare record systems. And notoriously, these systems don't like talking to each other. The model of approach for handling genetic data within these EHRs traditionally has been, oh, just give us the data. Give us the data, we'll handle it, we'll sort it out. Within the NHS, our sense is that that is not a scalable approach. We need a more interoperable solution. And what we've done as part of the NHS England Network of Excellence for Pharmacogenetics is to separate the data from the application. So to develop a clinical data repository that at the moment we term Progress RX. And the idea is that is uploaded from the different GLHs around the country, and then it's converted into a data standard. And data standards in pharmacogenetics, if we are going to deliver that interoperable and scalable solution, are absolutely fundamental. And although the nomenclature exists for pharmacogenetics, a data standard for storage, persistence, and messaging of that data does not. There's been some attempts to do it, but this needs to be developed. And so we have a program looking at implementing this in clinical practice. In phase 1, because we've created this structured data, people can go in, interact with that data, see the results. But in phase 2 of our work, which we're going into at the moment, we're hoping to push that data, or rather the data, we're hoping to push that prescribing advice out into electronic healthcare records. So when a clinician goes to prescribe a certain medicine, let's say clopidogrel, they will see a pop-up within their record if they are a poor metabolizer for CYP2C19, let's say. And so that's the architecture that we think needs to be developed. What we've done as part of phase 1 within the particular trial that we're testing this within is to use a system called Geno, which was developed in-house in Manchester. Essentially, it's linked to the NHS spine. You can search for your patients of interest, and they were recruited to our study, which is called Progress. You see here they were recruited because they were prescribed citalopram. They're an ultra-rapid metabolizer for CYP2C19. You see clear and concise guidance, which has been co-developed with the clinicians who are actually using this system. And because we have done a panel test, you can, in theory, reuse that data again over time. So let's say the patient presents a year later and needs a statin. Well, they've got normal SLCO1B1 function, so you can give a normal statin dose. But this serves a purpose for now, and we've recruited around 250 patients to the study, and the feedback on the system has been positive, but ultimately, that's not what we want to do in practice. We know that if this is going to be delivered as a routine intervention, as the data from the IPTIP interim analysis showed, we need to make this, the barrier to the use of this data, very low. And asking clinicians to go out and access an external system, we just know will not work in practice. And so by storing this data within a cloud-based repository, using data standards, what it means is that you become agnostic of your genotyping platform, and you become agnostic of your EHR provider. And that hopefully will allow that scalability and that interoperability. And we've had very positive relations with First Data Bank, Epic, and EMIS, and this has been tested in practice, and we've just gone live this week at one of our sites with notifications in the primary care electronic healthcare record. So we've demonstrated that these patterns can work. And so what a clinician will see, this patient has been prescribed codeine, but they're a poor metabolizer, and sorry, it doesn't project overly well, but essentially they're a poor metabolizer for CYP2D6, meaning they've got reduced morphine formation, and so you should avoid codeine. That is what the type of thing that our clinicians will now see within their electronic healthcare record. This is all being trialed within the Progress study, so I'm not going to go into detail on the Progress study at the moment, but QR code to a video discussing the study. What we've managed to do is to optimize that turnaround time. So from the patient being seen by their healthcare professional, GP or pharmacist, they're recruited to the study if they're prescribed an opioid, an antidepressant, a statin, or a PPI. We're able to get a 7-day turnaround to get those results back in practice. And we've been able to do that because we have virtually no bioinformatic input within the laboratory based on the systems we're using. And so far, we've seen a change to prescribing in around a quarter of participants, and that work is ongoing. And so a bit more detail on the technical architecture, which we've developed with clinical architects.